Okay, so welcome back to this next video in which we are discussing the digestion and absorption of fats. Okay, so we've now discussed the digestion of fats. What we now want to discuss is the absorption of fats. Okay, now before we discuss the absorption of fats, what I want to do is discuss the histology of the small intestinal wall. Okay, so I want to take a cross-section across the small intestine wall and have a look at what we would see. Okay, right, so let's have a look at the structure of the small intestinal wall, because basically a good understanding of this is foundational to understanding the absorption process. Okay, so basically, what you find is these projections of the stomach wall outwards, okay, which are called villi, okay, and they're little projections of epithelial cells, in fact. So, let me show this. So, this is a villus here, okay, and then basically what will happen is you don't only have these evaginations outwards of epithelial cells, you also have invaginations inwards. Okay, so in between intestinal villi, you're also going to have invaginations inwards, like so. Okay, and these are called crypts of Lieberkühn, or less dramatically, they're also called intestinal glands. Okay, so then it will go on to the next villus here. So, basically, these are the epithelial cells which line the surface of the small intestinal wall. Okay, so we have epithelial cell after epithelial cell after epithelial cell. And the type of epithelium that you have lining the small intestinal wall is a simple columnar epithelium. Now, what does that mean? Well, basically, the simple refers to the fact that it's a monolayer of cells. You don't have multiple layers of cells, and you don't have it even looking as though it's multiple layers of cells. Some epithelia can actually look as though they're multiple layers of cells, even though they aren't. Okay, right. So this is a simple columnar epithelium. The columnar means that the epithelial cells themselves are quite tall, okay, so they're column-like. Okay, so this is a simple columnar epithelium. Now, let's have a look at these epithelial cells in a bit more detail. Okay, so if we pull one of these out and have a look at its structure in more detail, we find that basically their apical surface is not just flat, instead they have a number of finger-like projections outwards. Okay, and these tiny little extensions outwards, these are called microvilli. Okay, so all over the larger villi that you have here, this actually is just a single villi, so we should call it a villus. The villus is the singular for villi, which is the plural. Okay, so all over this villus, all of these columnar epithelial cells, which by the way are also known as enterocytes, so the epithelial cells which line the small intestinal wall are also called enterocytes. Entero means pertaining to the intestine, cyte means cell, so these are just the cells of the intestine basically. Okay, so all of these enterocytes have on their apical surface little microvilli like so. Okay, and this little layer of microvilli that you have covering the larger villi is what's known as the brush border. So all of this, basically when you look at it underneath the microscope, all it looks like is a little fuzzy region basically. That's all you can make out. So people looking at it down the microscope called it the brush border basically. Okay, right. So, what else should we add to this diagram? Well, we should also add the label of what this thing is. So this is an intestinal gland, okay? So it has cells which secrete gut hormones, basically. But more important, more importantly to us, what I want to mention is that at the bottom of these intestinal glands, or their other names, are crypts of Lieberkühn. Okay, so crypts of Lieberkühn. Lipids is the other name for one of these. Okay, basically at the bottom of these crypts of Lipids, and that should have a funny little symbol like that on the top of it. Basically at the bottom of these crypts of Lipids, you have stem cells. So these cells that I'm going to highlight now in blue at the bottom of the crypts of Lipids, these are stem cells basically, and basically these stem cells are here to divide and produce new enterocytes. So these stem cells will be dividing and producing new enterocytes, and the idea basically is that these new enterocytes that you're producing from these stem cells, they will gradually make their way up the sides of the crypts of Lipids, then they'll make their way up the sides of the villus, and eventually when they get near the top they'll just sort of fall off into the intestine. So basically the epithelium of your small intestine is not constant. It's a continuous cycle basically. You're continuously producing new enterocytes that are going to cover the surface of your intestine, and they're continuously being passed out into the lumen of the intestine where they will be removed and excreted within the feces. Okay, so your epithelium of the small intestine is basically not a static structure, but it's continuously being rejuvenated, basically. Now, the next thing I want to show is what are all of these enterocytes sitting on? Well, basically all epithelia, no matter which epithelium you're talking about, all epithelia have basically what is known as a basement membrane underneath them. And basically, this basement membrane is a support for the cells. It is a protein meshwork that mainly consists of collagen, but also has other proteins such as fibrillin and laminins within it. And basically, all of the columnar epithelial cells are going to be attached onto this basement membrane, and that's what holds them in position. Okay, so this in turquoise here, this represents the basement membrane, and as I say, all epithelia have basement membranes. Now, the layer underneath the basement membrane is a layer called the lamina propria, which is a layer of connective tissue. Now, I won't show that yet because it's quite difficult to show that. Okay, it's easier first to show the layer underneath the lamina propria, which is called the muscularis mucosae, and then come back to the lamina propria once we've discussed the muscularis mucosae. So this layer here is going to be the muscularis mucosae. Okay, so this means the muscle layer, that's what muscularis means, and mucosae means of the mucosa. Okay, now I'll tell you what the mucosa is in a moment. Okay, so this muscularis mucosae consists of loads and loads of smooth muscle cells. Okay, so you have lots of smooth muscle cells connected to one another. And it's not a particularly thick layer of smooth muscle cells, it's a very thin layer of smooth muscle cells compared to the main proper muscle layers of the small intestine, which we'll see in a moment. Those are called the muscularis propria. Okay, so this isn't the main layer of smooth muscle cells that you have surrounding the small intestine, but it is a layer that surrounds the epithelial layer, the most outer layer, the closest layer to the lumen. Okay, so this is the muscularis mucosae, which is a thin layer of smooth muscle cells. And basically, we can now show the lamina propria. The lamina propria is all of this gap between the basement membrane and the muscularis mucosae beneath. So what you can hopefully appreciate is that the lamina propria has varying thicknesses. Okay, so if we're talking about a little bit where there is a crypt of Lipids, such as here, then the lamina propria is going to be very thin because you've got this invagination of the epithelial cells and the basement membrane inwards towards the muscularis mucosae, so that hugely reduces the thickness of the lamina propria here. Whereas if we're talking about a portion where we have a intestinal villus, like so, we can see that the thickness of the lamina propria is hugely increased there. So basically, lamina propria will have differing thicknesses depending on whether we're looking at a portion which has an intestinal villus, for instance, or a crypt of Lipids. Okay, so all of this all of this space between the basement membrane and the muscularis mucosae, this is called the lamina propria. And basically it's just a layer of loads and loads of connective tissue. Okay, so lots and lots of collagen. Now I might just sort of colour it in a sort of yellow colour, like so. But actually the yellow colour will get in the way of what I want to draw later. So I'll draw a bit of it in yellow, but I'm going to leave this bit free of yellow. Okay, right, because the lamina propria will have some important structures in it that I haven't yet shown, which are the blood vessels. And the reason I haven't yet shown them is because they're going to come from bigger blood vessels, which will come from the layer underneath the muscularis mucosae, which I'm about to discuss. Now, before we move on to that layer, I just want to now finally tell you what the mucosa is. So basically, the mucosa is a name that means all of the 4 layers that we've seen so far. So it means the enterocytes, these simple columnar epithelial cells. It means the basement membrane that the enterocytes are sitting on. It means the lamina propria of connective tissue underneath the basement membrane, and it also means the muscularis mucosae, which is underneath the lamina propria. So all these 4 layers together is known as the mucosa, or the intestinal mucosa in full. Right, so underneath the muscularis mucosae then you have another layer of connective tissue. Okay, so we have another layer underneath the muscularis mucosae, and this now is called the submucosa, and we can now understand why it's called the submucosa, because the mucosa is everything that it was above this. So this is the layer that's underneath the mucosa, hence its name, the submucosa. So this is another layer of connective tissue, similar to the lamina propria, and it basically is going to contain bigger blood vessels than the lamina propria. So it will contain the big arterioles and the big venules, and basically only small branches will come off these big arterioles and big venules, and those will penetrate the muscularis mucosae and go up into the lamina propria and then branch into capillaries, which will service the epithelial cells. So, for instance, here is a smaller arteriole coming off our large arteriole, okay, like so. And then, of course, this will branch into loads of capillaries, and those capillaries will collect the nutrients that is coming in from the intestinal lumen through these enterocytes, and then we have a venule which will then bring the blood back to our large venule down in the submucosa. Okay, so every intestinal villus will have an arteriole and a venule within it, like so. Now, there's another important vascular structure that is going to be really important within the small intestine, and these are lymphatic blood vessels. So I will show these in orange, although that's not a conventional colour to show lymphatic blood vessels. So basically every single one of these intestinal villi is going to have within it a special vascular vessel, which is called a lacteal basically, which will drain into one of these bigger lymphatic vessels down in the submucosa. Okay, so this lymphatic vessel which went up with the arterioles and the venules into the villus like so, this is known as a lacteal. Sometimes people will call these central lacteals. Okay, so every single villus will have a central lacteal, and the reason they're called lacteals is after milk basically. It should remind you of lactose, which is the key sugar within milk. Okay, and basically it's because the fluid which these lacteals have within them contains a lot of fat molecules and looks like milk basically. It's a white sort of creamy liquid that looks like milk, so that's why these are called lacteals. Okay, they're going to drain into bigger lymphatic vessels which will be within the submucosa down here. Now, for our purposes, we don't actually need to go any further, but there's hardly anything left over, so we might as well just, for completion, finish off the structure of the intestinal wall. Okay, so the next layer outside of the submucosa is then called the proper muscle layer. Okay, so this is the proper muscle layer that's going to contract and move food boluses along the intestine. Okay, so this is called muscularis, which means muscle layer, and then it's propria. So basically, muscularis propria can be split into two separate layers. It can be split into a circular layer and then also a longitudinal layer. And what is the difference between these two layers? Well, basically it's the orientation of the smooth muscle fibers which are in these two layers. Basically, if we draw a little picture of the intestine like so. So here's our small intestine here. Basically, the smooth muscle fibers that are in the circular portion will move longitudinally. So they will have their fibers in the longitudinal direction. Okay, so they'll be moving around the circumference of the small intestine, basically. Okay, whereas the fibers which are in the longitudinal portion of muscularis propria, they will have their fibers oriented in the longitudinal direction, so they'll be oriented along the length of the small intestine. So if I draw this really vividly, here is a smooth muscle fiber that will be in the circular portion, and here's a smooth muscle fiber that will be in the longitudinal portion. So they're the same sort of smooth muscle fibers. There's not any difference between them. All that's different between them is which orientation they're in. Okay, and then the final layer of the intestinal wall is basically a monolayer of cells. Okay, like so. And this is basically called the serosa. Now, this basically is the peritoneal covering that you have around the small intestine. So basically, the small intestine is suspended within the peritoneum, okay, by a mesentery. And when it was pushed into the peritoneum, or at least you can imagine it being pushed into the wall of the peritoneum, it gets a covering of peritoneal cells over it. And this is what we have here. So this is the serosa. Okay, so let me just discuss that with you in a bit more detail. So basically, if you imagine the peritoneal cavity as being like a balloon, okay, so how should I draw this? I'd like to draw a 3D picture if possible. So imagine the peritoneal cavity as kind of like this cubic balloon here, okay? And basically the surface of the balloon represents the peritoneal membrane. Okay, and then the contents of the balloon, instead of having the balloon filled up with air, imagine having it filled up with a fluid, and that fluid is going to be the peritoneal fluid. Now basically, even though it looks as though the intestinal organs are within the peritoneal cavity, they are not actually within the peritoneal cavity. None of them are actually in contact with the peritoneal fluid that is inside of the peritoneal membrane. Instead, what happens is basically you are going to take your intestine and you're going to sort of push it into the wall of the peritoneum. Okay, so if you imagine taking your finger and pushing it into the side of the wall of the peritoneum, and your finger just sort of invaginates into the peritoneal membrane, okay? So you'll just let this membrane be extremely sort of fluid, and you can distort it around your finger. And then what you're effectively going to get is something that looks like so, okay? So you've got this sort of finger shape here where you've stuck your finger in, and your finger is aligned by the peritoneal membrane, okay, but your finger has not punctured the membrane. It hasn't gone into the peritoneal fluid, okay? But now your finger looks as though it's in the peritoneal cavity, and it is covered by a layer of the peritoneal membrane. That is essentially what has happened to the intestine. It's sort of been pushed into the peritoneal membrane, and it looks as though it's dangling within the peritoneal cavity, but in fact it's actually outside of it, because you've always got this peritoneal membrane covering that's separating the intestinal organs from the peritoneal fluid. Okay, right, so that's basically the concept of everything being covered by a layer of peritoneal membrane. Okay, right, so we were doing this histology so that we can actually understand digestion. Okay, so basically these tiny little mixed micelles here are going to bring the products of digestion, so the free fatty acids, the 2 monoacyl glycerols, the lysophospholipid and the cholesterol molecules. They're going to bring them to these enterocytes here, and they're sort of going to go down these little gaps between the microvilli, basically. And then what will happen is when they go down these little gaps and they're surrounded by membrane, these molecules, which are all extremely lipid soluble, will end up leaving the mixed micelles and going into the membrane of these enterocytes. So the products are all now leaving the mixed micelles and going into the enterocytes. Now what's going to happen after that? Well, basically, most of these lipid molecules will now go onto the smooth endoplasmic reticulum, which is within the enterocyte. So let's get another piece of paper here. So, basically, if we draw a picture of our enterocyte here, so here it is, and it has on its apical surface these microvilli, which have now done their part. They have brought the products of digestion into the cell. What's now going to happen is these lipid molecules will go onto the smooth endoplasmic reticulum. Okay, so I'll draw this here. So this this membrane-bound organelle, this is going to represent the smooth endoplasmic reticulum. And basically what's now going to happen in the smooth endoplasmic reticulum is that we are going to reverse what happened in digestion. Okay, so from free fatty acids and 2 monoacyl glycerol molecules, we are going to resynthesize triacyl glycerol molecules. So let me draw this. So here are 2 free fatty acid molecules, and here is our 2 monoacyl glycerol molecule. Okay, so this is 2 mag, and these are 3 fatty acid molecules, and I'll colour it in because it looks scary at the moment, whereas once we've got some colour on, we know instantly what everything is. Okay, so here is our 2 monoacyl glycerol, and here are 2 free fatty acid molecules, and basically in the smooth endoplasmic reticulum, which by the way is often abbreviated to SER, like so, you're going to resynthesize these together to make triacyl glycerols again. So you're undoing the work of digestion, basically. So here we get our triacyl glycerols reformed here. Okay, so here's our glycerol molecule, and here's our 3 long-chain carboxylic acids linked off that. Okay, right. I think we'll call it there for this video, and we'll continue our discussion in the next video.